On January 17, 2011, Professor Don Huber wrote a confidential letter to U.S. Agriculture Secretary Tom Vilsack, alerting him that a team of scientists had discovered an unknown pathogen. The new pathogen was found in high populations in U.S. corn and soybean crops, specifically crops that have been genetically modified to be resistant to the chemical glyphosate and the #1 selling weed killer Roundup. The new organism was linked to an epidemic of plant diseases that had spread across Midwestern fields and found in cattle and dairy herds that were experiencing infertility and spontaneous abortion rates as high as 45%. Professor Don Huber is an internationally recognized plant pathologist and professor emeritus at Purdue University. For the past 40 years, he spent his career as a scientist working in professional and military agencies that evaluate and prepare for natural and man-made biological threats, including germ warfare and disease outbreaks. As a plant pathologist, Don Huber is among the most qualified scientists in the world to examine such a serious threat to America's food supply.
I'm curious why you wrote Secretary Vilsack the letter prior to the research being published.
He had a decision to make relative to Roundup Ready alfalfa. We didn't have answers that we needed in this area at the time, and when you start applying glyphosate directly to alfalfa, is it going to be similar to increasing the presence of the organism and the possibility in the alfalfa like it does in corn? If that's the case, we could very well lose the most critically important forage crop that we have for our animals. And it was a request for that funding so that we could get the answers and for him to delay a decision until the answers could be obtained. The intensity of the problem is such that we can't wait the 3 to 5 years to develop all of that research and then finally get it published.
These are preliminary results, findings that the science is still being conducted.
The animal scientists, veterinarians, it's not preliminary for them. They've very thoroughly documented what the cause of the problems are in the infertility and the spontaneous abortion situation. I had a report last week of 3 new herds one in South Dakota and 2 in Iowa. They're running 15% infertility, regardless of how many times that they tried. They couldn't get the heifers to settle. And then on top of that, they were running 35% spontaneous abortion. Well, there's no way to maintain an economic viability in that type of situation, and that's what growers are finding. But in trying to find out what was causing our animal producers to go out of business, literally, because of the high mortality and inability to reproduce, that they started looking at the feed. Said, how are they being infected? Where's this organism coming from? They then traced it to soybean meal initially. Started looking at how the soybeans would be infected. And you can find it in soil, the organism. You find it internal in the mycelium of the sudden death syndrome fungus. You also find extremely high titers or high populations of this organism in soybean sudden death infected plants.
Can you tell us a little bit about the pathogen specifically?
The organism can be seen only in an electron microscope. Very small, about the size of a small virus. It's culturable, in other words, it can be grown in culture, which we don't find with a virus. It's not a prion, it's not a mycoplasma. Very small, does replicate, reproduce itself. Very common hitchhiker with other organisms. I mentioned that it's been found in the mycelium, inside fungi. Grows very well in the presence of bacteria. It's infectious to cattle, pigs, horses, poultry, It will kill a fertilized egg in 24 to 48 hours. Animal scientists have done the Koch's postulates, that scientific process of establishing cause-disease relationship, but it's just an ultra-microscopic organism that's capable of reproduction, so it's different from a virus, very compatible with other organisms, appears to be very common in the environment, but for some reason much higher association and colonization or population when we have the glyphosate interaction or the Roundup Ready, and especially where we see those diseases that are increased by the glyphosate. We see also the higher population of this organism.
Why are the issues with glyphosate relevant to the discussion about genetically modified crops?
Glyphosate is the chemical name for that compound that is attributed for herbicidal action. The Roundup is a formulated product that includes other components besides just the glyphosate chemistry in order to increase its penetration into plant cells or its activity as an herbicide. When you have a genetically engineered crop that's resistant to glyphosate, it not only permits the wider use of the glyphosate, the extended exposure to it, but it also permits then the application of glyphosate directly on that crop, which many times we're consuming. What you see is that after a period of time of continued use of just one herbicide and increasing rates of that herbicide, which have been very dramatic in the last 10 years, that you see pretty soon that entire groups of organisms just cease to exist in the soil. Now, you don't have a void in the soil, so that if you pull one group out, you have another group that comes in. You have this very dramatic change in the soil microflora and that soil biota that affects these soil-borne diseases very dramatically. And that's because glyphosate is not only a very powerful herbicide, but it's also a very strong biocide, very selective in the soil, but it stimulates some of our soil-borne diseases, and it also changes the soil microflora that affects nutrient availability then for the crops. One of those critical components in that soil microflora that disappears with glyphosate or are severely suppressed are those organisms that make manganese available for the plant. When manganese is reduced in the plant, again, it becomes very susceptible to this pathogen. And so you have a biological component that is very powerful because of the powerful biocidal effects of glyphosate, and you also have a direct effect of glyphosate as a very strong chelating agent. Now, by chelator, we mean that's a chemical that can grab onto another micronutrient or other mineral element that's essential for a plant and immobilize it. And when that's immobilized, then the plant goes into a deficiency or reduced efficiency, so it becomes much more susceptible then for take-all disease as well as many other diseases.
The overuse of glyphosate or the reliance on glyphosate in the past 30 years, you're saying has created a system for soil microorganisms where certain microorganisms have been killed off or no longer prevalent, and it's allowed, inversely, it's allowed a certain other microorganisms, fungal, other type of bacteria to be present. The question is, if, in doing root damage is and inhibiting certain micronutrients in plant absorption, is this actually decreasing the amount of the nutrient value of some type of food crops that we're feeding to livestock, but also to human, like corn and soybean products?
Yes, in fact, my first real attention to that item was from a phone call from a veterinarian in Iowa. And he had some clients that had very high disease levels in spite of all the drugs that he was trying to give them and trying to keep them in a productive mode. And he felt like it was